Hello, I'm Dan Dever from the University of Pittsburgh. And I'd like to start by thanking the organizers of this session for giving me the opportunity to report on some of our preliminary data on the role and functional localization of KCNQ and the K2P or 2-pore potassium channels in modulating chloride secretion across primary human bronchial epithelial cells. This work has all been done in collaboration with Bob Bridges and his team, including Allie Vetter and Matt Green at the Chicago Medical School. I have no disclosures to report for this presentation. So you've all seen the basic model of chloride secretion, of course, many times. But what I'd like to focus on is the role of the potassium channels in maintaining the electrochemical driving force for chloride exit across the apical membrane. My lab has been instrumental in understanding the role of K-calcium 3.1, or the calcium-activated potassium channel, in this process. Indeed, we demonstrated now some 20 years ago that activation of this channel with pharmacological agents, including DCEBIO, could stimulate a large and sustained chloride secretory response across human airway, including in K3 cells and in primary cultures of human bronchial epithelia. However, other labs, including the labs of Cowley, Krindler, and Berkman, have identified additional channels that bear a role in maintaining chloride secretion. These include the KCNQ channels, including KCNQ1, which is known for many years to play a role based on its inhibition by 293B, a specific inhibitor of this channel. But these investigators also identified KCNQ3 and KCNQ5 based on PCR and antibody work and demonstrated that they played a role in chloride secretion across human airway, again in both K3s and HBEs using blocker pharmacology. These labs also reported a role for the K2P channels, including the TWIC channels, TREC1, and TASC2. And again, these channels were identified based on both immunoblots, immunofluorescence, and PCR work. And they were shown to play a role based on their blocker pharmacology. So relevant to our work, these labs, as indicated here in the bottom right, demonstrated by both immunofluorescence and electrophysiology that the 2-pore K channels were located exclusively to the apical membrane. And again, this was in both K3s and primary cultures of HBEs. KCNQ1 was located to the apical membrane as well as the basolateral membrane. KCNQ3 was located exclusively apically, and KCNQ5 was shown to be both apically and laterally. So in our case, we wanted to confirm we could identify these channels, and so after our electrophysiology experiments were done, we isolated proteins from the filters. Then Allie Vetter did these immunoblots to show expression first of K-calcium 3.1, which is a channel that we use as a control as we've studied a great deal. She also was able to identify TASC2, TREC1, KCNQ3, and KCNQ5 based on proteins running at the apparent molecular weight consistent with these channels. So we were able to confirm expression of these channels. The first thing we wanted to do then was to do some electrophysiology experiments. And for those, we used the following methods. We used primary human bronchial epithelial cells. These were grown in transwell filters in air-liquid interface for 5 or more weeks. Experiments were performed on a 24-channel MTEC and bicarbonate-free buffer to assess changes in the chloride equivalent current. The blockers for KCNQ, including 293 and XE991, as well as the blockers for the 2-pore channels or K2P channel, bupivacaine, were dissolved in DMSO, and these were added to both membranes given their lipophilic nature. We were then able to estimate the effects of these blockers on the apical and basolateral membrane resistances, that is RA and RB, using impedance analysis. In our case, all of the F5-way Del CFTR HBEs were incubated in C18 corrector for 24 hours in order to induce apical expression of this channel. So we started by looking at K-calcium 3.1, as this is a channel we've studied a great deal. So I should say in all of our recordings, I've eliminated the benzamyl-induced block of the sodium current so that it's easier to appreciate the chloride currents observed. So as you can see, forskolin, of course, causes a nice increase in current as expected when added. And looking at our impedance spectra and fitting, we could see an apical membrane resistance of about 28 ohm centimeters squared and a basolateral resistance of approximately 1,000 ohm centimeters squared. When we then added DCEBIO, we got a large increase in current, as we previously demonstrated some 20 years ago. And this resulted in a small decrease in the apical membrane resistance but more importantly, a dramatic decrease of over 500 ohms in the basolateral membrane resistance, which is consistent with activation of K-calcium 3.1 on the basolateral membrane as we previously described. We then added 293B, which is a specific inhibitor of KCNQ1. This produced a rather small decrease in current, and we saw no change in RA or RB with this addition. In contrast, when we added XE991, which will inhibit also KCNQ3 and KCNQ5, we saw, again, a very small change in the apical membrane resistance, but in this case, an approximately 110 ohm increase in the basolateral resistance, which is consistent with this channel being in the basolateral membrane and being blocked. Addition of TRAM34, which is a specific inhibitor of K-calcium 3.1, produced a nearly complete inhibition of the current induced by Forsglin and DCBIO. And that, again, had very little effect on the apical membrane resistance, but increased the basolateral resistance by nearly 500 ohm centimeters squared. This is consistent, again, with K-calcium 3.1 being expressed on the basolateral cell surface and being blocked by TRAM34. So based on this, we conclude that K-calcium 3.1 is, in fact, a basolateral conductance, similar to what has been described before. And it also provides some of our first evidence that KCNQ channels are also located on the basolateral membrane of primary cultures of human epithelia expressing the wild-type CFTR. In the rest of our studies, then, we concentrated on the KCNQ and K2P channels. So, again, in this case, we looked at a wild-type CFTR expressing HBE. And here, we fitted our data in the presence of benzamyl and had an apical membrane resistance of approximately 370 ohm centimeters squared, and again, a basolateral resistance of approximately 1,000 ohm centimeters squared. Addition of Forsglin stimulated a current response as expected, and this caused a significant decrease in both RA to 24 ohm centimeters squared, as well as a large decrease in RB to 750 ohm centimeters squared. This is consistent with what's been known for many decades now, that addition of Forsglin activates both an apical membrane chloride conductance, which we now know to be CFTR, as well as a basolateral membrane K conductance, which are the KCNQ family members. Addition of XE991, which again blocks KCNQ3 and 5, had no effect on the apical membrane resistance at all, but increased the basolateral membrane resistance approximately 70 ohm centimeters squared. Addition of bupivacaine subsequent to that again had no effect on apical membrane resistance, but further increased the basolateral resistance by approximately 80 ohm centimeters squared. So again, this increase in basolateral membrane resistance following block of an ion channel is consistent with these channels being expressed on the basolateral surface. So our conclusion from these data is that both KCNQ and the two pore channels are located on the basolateral side under our conditions. We then reversed the order of addition of these blockers to see if that would make any difference. Again, Forsglin stimulated a good current response. We had similar apical and basolateral resistances to what we saw before. In this case, addition of bupivacaine first to block the K2P channels, again, had no effect on the apical resistance, but did increase the basolateral resistance by approximately 160 ohm centimeters squared. The further addition of XE991 to block KCNQ3 and 5 further increased the basolateral resistance by approximately 130 ohm centimeters squared, where again it had no effect on the apical membrane resistance. So again, the addition of these blockers causing an increase in the basolateral resistance is consistent with both of these classes of channels being functionally located to the basolateral membrane in wild-type CFTR expressing HBEs. We then turned our attention to F508 del homozygous experiments, and is shown here. In this case, again, we incubated overnight in C18. We then stimulated with Forsglin plus VX770 as a potentiator of the F508 del CFTR, which stimulated a significant increase in current as expected. XE991 and bupivacaine both inhibited the current following this activation by Forsglin plus VX770. But in sharp contrast to what we observed on the wild-type CFTR, in this case, you'll notice that the apical membrane resistance in both donor 1 and donor number 2 actually increased. So the resistance in donor number 1 went from 420 ohm centimeters squared, it increased by 200 ohm centimeters squared following block of the KCNQ channels, and increased by an additional 160 ohm centimeters squared following block of the K2P channels. In donor number 2, we saw similar effects where the combined increase in resistance was approximately 225 ohms centimeters squared on the apical membrane. The basolateral resistance paradoxically actually decreased. It decreased more in donor number 1 than it did in donor number 2, but they're qualitatively similar results. So we interpret these results to indicate that the apical membrane contains the functional KCNQ and K2P channels, which is reflected by blocking these channels and causing an increase in the apical membrane resistance. The decrease in basolateral resistance may in fact be the cells attempting to recover following block of the apical membrane, although that requires more clarification. But in any event, a decrease in resistance on the basal membrane cannot account for the inhibition of current that we observed. So based on that, we conclude that the KCNQ and K2P channels are actually located functionally in the apical membrane in F508del homozygous CFTR-expressing human bronchial epithelial cells, in stark contrast to what we observed in the wild-type CFTR-expressing cells. I should say we did these experiments in addition with C17 as a corrector. We also did experiments using a different potentiator rather than VX770. And in more than 100 filters from 2 different donors on multiple different days, we always saw an increase in the apical membrane resistance when we blocked either KCNQ or K2P channels. Next, we looked at the gating mutation S549N to see whether or not these channels were apically or basolaterally located. So this has an F508del background, and so initially we did experiments in the absence of C18, is shown here. This way we can study S549N in complete isolation from the F508del, as it would not traffic to the membrane under these conditions. We still see a small response to forskolin, and again, VX770 as a potentiator significantly increases the current. This is blocked by both XE991 and bupivacaine, and is shown here in the right. You can see that the basolateral membrane resistance is actually increasing by approximately 70 ohm centimeters squared. At this point, we have a relatively small number of studies being done with n equals 6. Again, the apical membrane resistance shows a small decrease, which cannot explain the decrease in current following addition of a potassium channel blocker. So this increase in basolateral resistance would suggest KCNQ channels are located, in this case, functionally on the basolateral side. Given the fact that while we saw a block with bupivacaine, but we saw really no change in resistance, we cannot at this point determine whether or not these K2P channels are also located on the basolateral surface, although that is our suspicion. We then did experiments in the presence of C18 to correct the F508del CFTR. In this case, the forskolin response and the VX770 response were larger than the absence of C18, consistent with expression of F508del at the plasma membrane under these conditions. Again, XE991 produced a significant block of this current. And again, the increase in resistance of approximately 140 ohm centimeters squared was all on the basolateral side. The apical membrane resistance, if anything, again, slightly decreased. While bupivacaine addition, again, blocked the current, it did not go below what we observed with XE991 transiently, and therefore we saw no change in either basolateral or apical resistance. But again, based on these results, we feel that we can place the functional KCNQ channels at the basolateral surface in this S549N gating mutation. The fact that C18 did not increase the response or switch it over to the apical membrane suggests that the F508del, in fact, is not a dominant phenotype. So in summary then, our impedance studies indicate functional KCNQ and 2-pore potassium channels are localized to the basolateral membrane during forskolin-stimulated chloride secretion in wild-type CFTR-expressing HBEs. We demonstrate for the first time a functional role for KCNQ and KTP channels in chloride secretion across both homozygous F508del patient samples, as well as in a gating mutant S549N. In sharp contrast to results on the wild-type CFTR HBEs, our impedance studies indicate that the functional KCNQ and KTP channels are localized to the apical membrane during forsplin plus VX770 stimulated chloride secretion in C18-corrected F508del homozygous CFTR-expressing HBEs. Again, in contrast, in the gating mutant S549N, our impedance studies suggest that at least the KCNQ channels are, in fact, again, appear functionally on the basolateral surface. And this is not altered by C18-mediated correction of the F508del CFTR. So what are the consequences of this localization of these channels? Well, increased functional KCNQ and KTP channels in the apical membrane of the F508del homozygous CFTR HBEs would further hyperpolarize the apical membrane relative to basolateral localization of these channels. That would then increase the electrochemical driving force for chloride exit across the apical membrane under these conditions. In addition, the apical localization of functional KCNQ and KTP channels in the F508del homozygous CFTR-HBEs would be anticipated to result in increased potassium exit across this apical membrane during chloride secretion. These results, in fact, could help explain the increased airway surface liquid potassium that has been previously reported by some labs. In future studies, we would like to examine the localization of KCNQ and KTP channels in F508del homozygous CFTR-HBEs following correction with the clinical compounds VX445 and VX661 to see if that makes a difference in terms of where we see these channels localized functionally. We'd also like to assess the localization of these channels by both IF and side biotinylations to determine if there's actually a silent pool of channels that are not being activated during our impedance studies, consistent with what other labs have reported with their IF data. Of course, we'd also like to carry out studies on additional patient samples and CFTR genotypes, including additional trafficking gating mutations, as well as following correction of premature termination codon mutations to determine the functional localization of the KCNQ and KTP channels. We would also then like to determine whether these effects have any effect on the airway surface liquid height. Finally, I would like to acknowledge my collaborators, Bob Bridges, who's been instrumental in this, and the people in his lab, Ali Vetter and Matt Green, as well as at the University of Pittsburgh, where Michael Meyerberg and Agnes Urban have begun to work with us prior to Agnes leaving to do some of these studies. And of course, these could not have been done without the generous funding from the CF Foundation. Thank you very much.
Hello, and welcome to this presentation in the ion transport in CF pathology section of this meeting. I'm Bruce Schultz, and for this virtual meeting, I'm coming to you from the beautiful Flint Hills and tall grass prairies near Manhattan, Kansas, where I'm a professor at Kansas State University. Before going on, I want to extend my sincere thanks to the CF Foundation for providing this venue for the presentation of outcomes of our work, and my sincere thanks to the organizers for inviting me to participate at this level. There are no relationships to disclose relative to this presentation. The title of the presentation is Bicarbonate Conductance Stimulated by Correctors and Potentiators Differ Across CFTR Variants. At this point, I would like to acknowledge the major contributions of my collaborators and co-workers, especially Bob Bridges at Rosalind Franklin University and all the folks that work in his lab there. Also to Dong-O Lee, who works in my lab here at Kansas State University. As we attempt to look at bicarbonate conductance in epithelia, we have to start off with some basic models and think about the contributions that they can provide, or the hypotheses that can be tested with each. The simplest CFTR-dependent cell model to account for chloride secretion has been with us for some time, proposed by the likes of Field, Frizzell, Boucher, and etc. And it's a fairly simple model with 3 items in the basolateral membrane that allow for membrane potential to be maintained, and for chloride loading to occur while maintaining sodium and potassium recycling back to the blood side or basolateral side. Stimulation of a G protein coupled receptor leads to activation of pKa and ultimately to phosphorylation of CFTR, and then allowing the exit of chloride down its electrochemical gradient. This simple model can be embellished very simply by the addition of a bicarbonate loading step, in this case NBC or SLC4A4, that allows for bicarbonate to accumulate in the cell above its electrochemical potential, and then with the activation of CFTR, if CFTR is in fact bicarbonate permeant, then bicarbonate can be secreted across the epithelium into the luminal compartment. More elegant models to account for bicarbonate secretion, CFTR-associated bicarbonate secretion, have been proposed, where we have, for example, here this chloride bicarbonate exchanger from the SLC26 transporter family, where you have chloride for bicarbonate exchange, chloride then recycling through the apical CFTR. Each of these models has certain strengths and weaknesses as they encompass or incorporate new observations. Critically, one of the shortcomings of this particular cell model is it fails to account for the very high bicarbonate concentration that can be seen in certain fluids such as pancreatic juice. And so additional work must be done to identify the bicarbonate secretion mechanisms that are present. Ultimately, the critical question is CFTR permeant to bicarbonate? And that bicarbonate is important to the CF field has been known for quite some time. As early as 1982, Paul Quinton reported that in CF sweat ducts, there was a difference in the anion gap compared to healthy sweat ducts. He attributed this to an absence or a reduction in bicarbonate secretion in CF individuals. However, these observations fell by the wayside as far as ongoing observations, and it wasn't until 20 years later that we became back interested in bicarbonate, and largely again with Paul Quinton's fixing us on this focus by stating in an editorial, simply fixing the chloride permeability defect by enhancing CFTR processing or by activating a supplemental chloride conductance may not be adequate therapy. He indicated in this editorial that bicarbonate and pH regulation could be critical in the therapy of CF patients. Ultimately, I thought that Bob did an elegant job of bringing a bunch of observations together in really distilling down what we need to know about bicarbonate and chloride permeability in cystic fibrosis. And the fundamental question that he asked was, is the anion selectivity ratio, the relative amount of conductance attributable to bicarbonate and chloride, does that differ depending upon what CFTR variant, and especially CF causing CFTR variants, are being assessed? And then building on this ratio of conductance due to bicarbonate relative to chloride, do drugs targeting the therapy of CF and targeting CFTR, do they differentially modify this selectivity ratio? Knowing that more recently we've heard or we've seen that different second messenger pathways can perhaps modify the chloride to bicarbonate conductance ratios, can these be targeted, and do they in fact change the conductance? And finally, are any of the above tissue phenomena tissue specific? For example, do we see different pathways or different regulation in the pancreas relative to airway, intestines, vas deferens, etc.? But in all of these things, we come back to one critical question. Can we measure bicarbonate conductance? And can we assess the relative conductance of chloride to bicarbonate? And so, starting with this rather elegant cell model, Dr. Bridges suggested some years ago that we step back and obtain a simpler model. And so he started by using the Fisher rat thyroid cell model. And this cell model is particularly important because when CFTR is expressed in these epithelial cells, it is trafficked to both the apical and to the basolateral cell membrane, so that now we can set up electro measurements putting in square wave steps in voltage, measuring the current, and using Ohm's law to determine the transepithelial conductance. And if we can pharmacologically identify CFTR in the membrane, then we can attribute the conductance measurements that we're making to CFTR associated conductance. And given that we can measure conductance relative to chloride, could we then measure chloride or conductance relative to bicarbonate? And with that in mind, we set up a system where we have three typical media that we use to assess conductance in pairwise experiments. One has a relatively high chloride concentration, 125 milliequivalents of chloride. At the other end of the spectrum, we have a low chloride medium that has 100 milliequivalents and is balanced osmotically with 50 millimolar mannitol. And between those, we have a medium that has the same chloride concentration as the low chloride, but the same ionic strength as the high chloride. So we have an increment of reduction of 25 milliequivalents per liter of chloride replaced by 25 milliequivalents of bicarbonate. So can we use this system then to assess CFTR-dependent anion conductance? So in our first set of experiments, we sought to validate the system, and we can measure conductance across FDR-T cells expressing wild-type CFTR and document that this conductance is proportional to the concentration of the conducting anion, in this case, chloride. So I'm going to walk through this image to describe the experimental protocol that we use that will be used in the next series of slides. On the left, we have a time course with a number of different conditions in which we establish a baseline conductance, then we expose the cells to forskolin, and we see this increase in conductance as shown on the ordinate going from here 0 to roughly 45 millisiemens per centimeter squared. We see this increase in conductance in response to forskolin. We then add and mix VX770 into the media, and again have this static media where we measure conductance over a period of time, then expose the cells to a CFTR selective inhibitor, either GlyH101 or INH172, to reduce the conductance and convince ourselves that we're in fact looking at a CFTR associated conductance. In this image, what we show is we start off with 139 milliequivalents per liter of chloride, which is the typical concentration in Kuhn's medium that is used to grow these cells. We then decrease the chloride concentration by 10% increments down to 73 milliequivalents per liter, and we see a proportional stepwise decrease in chloride conductance, or in transepithelial conductance in these monolayers. When we add VX770, there is an increment in conductance, but we still have a stepwise change in conductance associated with the anion concentration. Shown on the right are bar charts that have the average conductance for each one of these different conditions in the presence of forskolin, and we see this stepwise decrease in conductance associated with anion concentration. We do the same for forskolin plus VX770. We see a higher overall conductance in each condition, and by subtraction, we get the increment in conductance that's attributable to VX770. I want to call your attention to the open circles here that is associated with 125 milliequivalents per liter of chloride, and the open triangles at 100 milliequivalents per liter. So those are the two concentrations of chloride that we will use then in the remaining experiments, in which we set out to determine, can we detect a bicarbonate conductance that's associated with CFTR? And the answer is yes, yes, we can. In cells expressing wild-type CFTR, we see a substantial forskolin-associated increase in conductance attributable to bicarbonate. So shown here in the dark gray symbols is low chloride, in the black symbols is high chloride, and we see almost superimposable after exposure to forskolin is the low chloride plus bicarbonate. So this would suggest that in these conditions, bicarbonate supports nearly as much conductance as chloride for this 25 milliequivalent increment. After exposure to VX770, we see a separation of the three media with high chloride, chloride plus bicarbonate, and low chloride, such that when we, by subtraction, determine the increment in conductance due to VX770, we see that the increment in bicarbonate-containing medium with 100 milliequivalents of chloride is actually less than 100 milliequivalents of chloride alone. So there could be some difference between forskolin as an activator and VX770 as a potentiator in the bicarbonate chloride conductance ratio that observed. But certainly to ferret this out, we'll have to do more experiments. We then built on this by asking, can we see similar phenomenon when we look at variants of CFTR? And the first variant examined was the F508 deleted variant. And the title really says it all. No or very little bicarbonate conductance is stimulated by forskolin or by VX770 across these cells expressing this common mutant form of CFTR. Shown in the graphs are four conditions: vehicle, a first-generation corrector, a second-generation corrector, and then the combination of the two correctors. And I want to point out straight away that there's a substantial difference in the ordinate scale. 4 millisiemens per centimeter squared, 8 for each of the correctors alone, and then all the way up to 60 millisiemens per centimeter squared for the combination of the two. When we look at the vehicle-treated cells that were exposed for about 12 hours, the signal is relatively low, and we can't differentiate a difference between the different media that were used. But with either the correctors, C17 or C18, we do see a distinct pattern, and that is that both forskolin and VX770 increase in a stepwise way the overall conductance that is observed across these cells expressing delta 508 CFTR. What is immediately obvious is that the light gray symbols are closer to the low chloride than they were with wild type CFTR. And in fact, in the presence of forskolin and VX770 with the C17, we see that the bicarbonate containing media is actually below the low chloride. And that's shown here on the right in the bar chart, that the bicarbonate containing medium actually has a lower increment in conductance in response to VX770 than the low chloride medium. The same pattern is seen with C18. So low chloride with bicarbonate is superimposable with low chloride is below low chloride. So clearly with these two correctors, we are not seeing an increment or evidence of a bicarbonate conductance for delta 508 CFTR. With both correctors, we see that there is an increment of bicarbonate conductance above the low chloride in both forskolin and forskolin plus VX770. However, the increment after VX770 is actually smaller than the increment with forskolin alone, such that it appears to be as small or less than that with low chloride. So overall, we're seeing a very distinctly different pattern in in bicarbonate conductance. There's not any evidence that really drives one to believe that there is substantial, or in some cases, even any bicarbonate conductance in the 508 deleted cells when exposed to either the first generation or the second generation corrector alone. So the next question we asked then was whether or not these observations could be extended to other variant forms of CFTR, and we looked at two of them. And in this case, rather than being trafficking mutations, we are looking at gating mutations. The first is the G551D, and the title of the slide says much. Substantial bicarbonate conductance is stimulated by forskolin and VX770 across these cells expressing 551D CFTR. The same pattern of figures is shown in the slide with vehicle, C17, C18, and the combination. Again, we have three different ordinate scales, 12, 20, and 30 millisiemens per square centimeter. What is immediately obvious is that, first of all, forskolin stimulates very little conductance in these cells expressing the gating mutation, but that VX770 greatly potentiates the response, and that we see that the bicarbonate containing medium, chloride, low chloride plus bicarbonate, is superimposable with the high chloride, in this case slightly above the high chloride, superimposable with. And so the G551D has very substantial bicarbonate conductance, and it's seen with all of the corrector combinations that were evaluated. So a distinctly different pattern from Delta 508. The next gating mutant that was examined is the S549R, and the pattern is slightly different than that seen for 551 that was just shown. Once again, we see differences in the ordinance scales 6, 20, 20, and 40 for this set of experiments. And with either vehicle or C17, we see that the conductance is superimposable with that of the high chloride. And so the bicarbonate is substituting for chloride almost on a one-for-one basis and contributing to overall conductance. In the presence of C18 or the combination of 17 and 18, the conductance does not parallel fully the high chloride condition, but is substantially closer to high chloride than to low chloride. So there is a substantial increment in conductance associated with bicarbonate being present in the medium. So from these sets of experiments, a series of conclusions can be made. First of all, the relatively simple FRT epithelial cell model is very well suited to assess CFTR-mediated anion conductance and to assess the anion conductance of CF-associated variants. We demonstrated by using various chloride concentrations that we could measure the chloride conductance, and the chloride and the transepithelial conductance was proportional to the permeant anion concentration. We then supplemented the low chloride condition with bicarbonate and showed an increment in bicarbonate or in transepithelial conductance in the wild type, the G551D, and the S549R CFTR expression systems. So we can clearly use this to determine the relative bicarbonate to chloride conductance. We have substantial differences between the variants that were observed. Finally, prototypical correctors enhanced the anion conductance for all of the variants that were assessed. And there were some differences in the pattern of the chloride to bicarbonate conductance that suggested that perhaps the correctors do have an effect on the conductance of the variant forms of CFTR. So with this in mind, one must consider that going forward, it might be important to very more closely assess bicarbonate secretion of the various forms of CFTR. And in the era of the triple corrector, going forward I believe that it's likely we will begin to see greater types of pathology or greater prevalence of pathologies that are associated with pH-dependent differences in the patients that have various different types of CFTR that they are expressing. With that, I would like to conclude by thanking you for your attention, and again, thanking my co-authors and contributors, Dr. Bridges and his lab, Doug Lee in my lab, and thanking the Foundation for their support and the Kansas State College of Veterinary Medicine for its support for this research.
Okay, good afternoon. I'd like to thank the organisers for the opportunity to speak this afternoon about some of the work that we've been doing looking at TMEM16A, a calcium-activated chloride channel, as an alternative pathway to enable both chloride and bicarbonate secretion in the airway epithelium of CF patients. Okay, my disclosures: I'm co-founder and stockholder of Enterprise Therapeutics, as well as being a full-time employee. So, as I said, TMEM16A, it's a calcium-activated chloride channel, but is able to conduct both chloride and bicarbonate ions. When calcium becomes elevated in the airway epithelium following, for example, stimulation of the P2Y2 receptor with endogenously produced ATP or UTP. It leads to opening of the channel and the flux of both of these anions. And because TMEM16A is obviously a completely independent channel to CFTR, it provides an opportunity, if we can enhance its activity, to treat all CF patients irrespective of their particular mutations in CFTR that they are carrying. So our approach here is to identify low molecular weight compounds that will potentiate the activity of TMEM16A such that when calcium becomes elevated in the cell, there is opening of the channel that starts sooner and lasts longer with the potentiator on board. So evidence that a calcium activated chloride channel is important in the airways, and particularly the airways of CF patients, initially came from Mike Knowles and Rick Boucher, who did a number of studies, and this is just one piece of data illustrating a raw data trace from a nasal potential difference study in a patient with CF. So initially, amiloride is perfused over the nasal mucosa, and we can see a depolarization as ENaC becomes switched off. They then switch to chloride-free conditions that essentially sensitize the system to be able to detect changes in chloride secretion across the mucosa, and with it being a CF patient, there's very little change, if any, in chloride secretion. That is until ATP is added into the nasal perfusate to stimulate the P2Y2 receptor, cause an increase in calcium within the nasal epithelium, and you can see under these conditions there's then a pronounced hyperpolarization of that nasal PD consistent with the activation of a calcium-dependent chloride channel. And recent studies, well, recent 2008, 3 papers came along indicating that TMEM16A was a calcium-activated chloride conductance, and suggesting that actually TMEM16A had a significant role to play in the calcium-activated chloride conductance in the airway epithelium. So our approach at the start of the drug discovery process was to ask whether or not, firstly, we could identify potent and effective TMEM16A potentiators. And I stress again here the mechanism. We're looking for potentiators of TMEM16A. We are not interested in compounds that will just cause a global elevation of intracellular calcium. We're looking really for compounds that specifically potentiate the activity of the channel when calcium becomes elevated endogenously. Assuming we can find TMEM16A potentiators, we can then go on to start essentially target validation and ask the question as to whether or not this can be relevant in CF-HBEs in terms of driving anion and therefore fluid secretion, and ultimately could this impact on mucus clearance in vivo. And then importantly, what might be the consequences from a safety perspective of enhancing TMEM16A function, not only in the lung but also in other organs where we know that TMEM16A is expressed. And these are the areas that I'll cover in the next few minutes of the talk. So the first question, could we identify TMEM16A potentiators? Yes, we undertook a high throughput screen, in fact multiple high throughput screens, and identified compounds like this low potency screening hit here. So what you can see, the black line is the control, illustrating the classic biophysical fingerprint of TMEM16A as we modulate the holding potential across these particular cells in the patch clamp assay. And then after multiple rounds of medicinal chemistry, a huge medicinal chemistry effort, compounds such as ETX001 were identified, which you can see on the right hand side when we plot the concentration response, are able to potently potentiate the activity of TMEM16A with an EC50 of around about 100 nanomolar, and enabling around a 3 to 4 fold enhancement of channel activity. And important to note here that although in these assays we are modifying voltage, what we are also doing is very tightly buffering intracellular calcium. So what we can be confident of is that these compounds are not causing an activation of TMEM16A by changing intracellular calcium levels. That really is very tightly buffered. So we can be confident from these types of experiments that the compounds are acting through a potentiation of the channel and not a direct activation. So what will compounds like ETX001 do in terms of epithelial cell responses, anion secretion, fluid secretion, and mucus clearance? So if you look at this raw data trace here, these are CF bronchial epithelial cells cultured at air-liquid interface. They've been IL-13 treated to upregulate TMEM16A expression to give us a good signal-to-noise ratio. And then what we can do at this point in the experiment is start adding increasing concentrations of UTP. And you can see that as the concentrations of UTP are increased, we see an increase in the short circuit current response as calcium is becoming elevated in the cells and opening TMEM16A. And we can plot the concentration response to UTP here on the right-hand side. What we can then do in matched cultures, exactly the same experiment, but at this point here, rather than adding vehicle, we actually add our TMEM16A potentiator, which in its own right causes a small step up in current. But then as we start adding our increasing concentrations of UTP, you can see the system is now sensitized, where previously a low concentration of UTP was not evoking a change in short circuit current. We're now seeing changes in current associated with those lower concentrations of UTP. And then as the concentrations increase, you can see we're seeing an enhancement of that anion secretory response. And if we plot this, for example, as area under the curve and plot that UTP concentration response in the presence of our TMEM16A potentiator, you can see a very clear enhanced efficacy at each one of those UTP concentrations. So consistent with the fact that our TMEM16A potentiator can enhance the activity of native TMEM16A in primary CF epithelial cells. And we've now undertaken these experiments in multiple different CF donor codes, including delta F508 homozygote and heterozygotes, and as well as patients with mutations that are insensitive to current CFTR repair. So consistent with the fact that these types of compounds will also work, have the opportunity to work in patients currently not suited to CFTR repair. So the next question becomes, well, could this actually be relevant, therefore, for fluid secretion into the airway lumen? And for these experiments, we've collaborated with Dr. Brian Button at the University of North Carolina. And what Brian's able to do is measure the height of the airway surface liquid and changes in height of airway surface liquid with various manipulations. So what Brian can do is stain the airway surface liquid, this very thin film of fluid that sits on top of the epithelium, with a red fluorescent dye. And then, for example, in this experiment, treat the cells for just 60 minutes with our TMEM16A potentiator, and at the end of which we can see around about a twofold increase in the height of the airway surface liquid, consistent with the compound having stimulated anion and therefore fluid secretion onto the airway mucosa. And we can quantify that on the right-hand side here. You can see a significant increase in airway surface liquid height with a 1 hour exposure to a TMEM16A potentiator. Then what's really cool is what Brian can do with this system is apply mechanical forces, and particularly shear stress, to these epithelial cells to mimic tidal breathing and the flow of air across the epithelium during breathing, which causes the release, the endogenous release of ATP that will then stimulate P2Y2 receptors on the apical surface of the epithelium, increase intracellular calcium, and lead to an activation of TMEM16A to flux chloride and bicarbonate, and thereby drive more fluid secretion. So after 4 hours of shear stress, the vehicle treated cells have also got an increase in airway surface liquid height because of this switching on of this ATP mechanism in the cultures. But those cultures that had also been pre-treated with the TMEM16A potentiator show a further enhancement of this fluid secretory response. So consistent with the fact that our TMEM16A potentiator can really enhance TMEM16A function even under conditions where calcium is being endogenously regulated through the mimicking of tidal breathing in these cultures. So the next question becomes whether this actually translates through to a meaningful mechanism in vivo. So to ask this question, we've collaborated with Dr. Juan Sabate at the Mount Sinai in Miami, where he has a sheep model of mucociliary clearance. So conscious sheep inhale radiolabeled particles and then are placed underneath a gamma camera, and you can monitor the rate at which these particles are moved up and out of the airway by mucociliary clearance. And in this particular experiment, at -4 hours, so over to the left-hand side somewhere, the sheep were administered either inhaled doses of vehicle or one of the 3 doses of our TMEM16A potentiator molecule. The sheep then wait 4 hours. Before then, at time 0, they're administered the radiolabeled particles, and we monitor the clearance out of the airway. And you can see that there is a dose-dependent enhancement of both the rate and the magnitude of mucus clearance in the sheep dosed with the TMEM16A potentiator, indicating that this does appear to be a relevant mechanism for enhancing mucus clearance in vivo. And the other thing to just point out is these are normal wild-type sheep that have normal CFTR function. So this looks like it's a mechanism that can operate on top of CFTR function. So although this obviously has to be tested in a clinical setting, there could be the potential that in a patient treated with CFTR repair, there's the opportunity for a further enhancement of mucus clearance on top of that with a TMEM16A potentiator. So finally, the question of safety and what might TMEM16A potentiation do both in other tissues in the lung and also other organ systems in the body. And if you look at the literature, there really is a growing body of data around functions of TMEM16A in a whole variety of different tissues. And I've just highlighted a couple of relatively recent papers that are obviously particularly relevant to those of us interested in respiratory diseases. This one here from Jason Rock's group indicating a role for TMEM16A in both mucin secretion and airway smooth muscle contraction in a murine model. Another one here indicating or suggesting that TMEM16A is indispensable for mucus secretion in the airways. And also further papers indicating that TMEM16A might actually be playing a role in actually driving the differentiation of the epithelium towards a goblet cell rich mucus hypersecreting phenotype. And based on these types of papers, it has more recently been suggested that actually blocking TMEM16A function may be of greater overall benefit in the terms of the treatment of respiratory diseases. So clearly an area that's both contentious and one that we need to really properly understand as we move into the clinical setting with these types of molecules. So the first studies we undertook were with Dr. Camille Eyre, and what Camille asked was whether or not she could find any evidence for TMEM16A potentiators like 001 enhancing mucin release from primary cultures of human bronchial epithelial cells, and in this case derived from CF patients. So cultures were exposed to the TMEM16A potentiator for 24 hours, and then MUC5AC and MUC5B levels were quantified on the airway mucosa, and Camille could see no evidence for any change in either MUC5AC or MUC5B release induced by the TMEM16A potentiator, despite a very clear effect of the positive control PMA. This was the case in both untreated cultures and also cultures that had been primed with 48 hours of exposure to interleukin 13. So IL-13 will both increase MUC5AC production within the epithelium, so effectively charge up the goblet cells, but also increase TMEM16A expression in its own right. So really giving ourselves the best opportunity to see a TMEM16A window if there's one to be seen. And under these conditions, again, Camille could see no evidence of an increase in either MUC5AC or MUC5B secretion induced by the TMEM16A potentiator. So no evidence in an in vitro setting that TMEM16A is playing a role in mucin secretion from primary human airway epithelial cells. So we then moved in vivo and collaborated with Dr. Burton Dickey and Dr. Michael Tuvin from the MD Anderson Center, where they are experts at looking at mucin release from the airways in vivo. And what we did for this experiment, we used a different TMEM16A potentiator because this one really shows good metabolic stability when administered to the animals, so we could achieve systemically relevant concentrations of a TMEM16A potentiator for an extended period of time out to at least 6 hours after dosing. And the dotted line here represents the ED50 for this particular compound on murine TMEM16A. So at both 0.3 and 3 mg per kg, we're seeing significant exposures above the ED50 for target engagement. So the model itself, the animals are dosed with interleukin-13 for several days to induce a goblet cell metaplasia. And then on the back of that goblet cell metaplasia, animals are administered the TMEM16A potentiator, and we can see no evidence of emptying of the goblet cells in this particular model. But the positive control for the system, inhaled ATP, caused around about a 50% emptying of the goblet cells. So the take-home message here is that in mice with a goblet cell metaplasia, we can see no evidence of a TMEM16A potentiator at levels way in excess of those required for target engagement having any effect on goblet cell emptying. And then finally, we have asked the question of smooth muscle contraction in vivo. And we've gone about this in two ways. Firstly, we've taken freshly isolated human bronchi, put them into wire myography systems, and put them in under conditions whereby we can see both a relaxant and a contractile effect of any given agent. And you can see in these studies, neither the TMEM16A potentiator 001 up to concentrations of 10 micromolar or ANI9, a potent TMEM16A blocker, were able to influence the tone in these freshly isolated human bronchial samples. We then moved in vivo and looked at conscious rats in a plasmography system, administered TMEM16A potentiator by direct intratracheal instillation to really achieve high doses directly into the airway. And under these conditions, we could see no effect on lung function, no evidence of any effect on airway smooth muscle in vivo. So in summary, yes, we've identified novel, potent and selective TMEM16A potentiators, and using these compounds, we can show that potentiating TMEM16A has significant effect on airway anion and fluid secretion, and translates through to enhancing mucus clearance in a large animal model of mucociliary clearance. And in terms of safety, although I've only really shown you a small fraction of the data that we've generated, we've certainly seen no effect of TMEM16 potentiators on lung function, mucus production, or release. And with these inhaled compounds also going into cardiovascular studies, no change in cardiovascular function or isolated human vessels. So finally, I'd like to acknowledge the input of a huge number of people on this project who put some tremendous effort in, and also to recognise the CF Trust and also the CF Foundation for their very much appreciated support on our work. So I'd be very happy to take any questions.
Hello everyone, my name is Gary Cutting and I'm going to be presented in this symposium on the topic of SLC 26A9 as a CF modifier. I'll just call it A9 from here on, repeating the whole sentence. My disclosures are shown here. And the question is, how do we become interested in SLC26A9? So that story actually begins with our search for modifiers of cystic fibrosis-related diabetes. It's an age-dependent complication that affects about 20% of adolescents, but a much larger fraction of adults with CF, so a very important complication associated with reduced survival and increased morbidity. There's high heritability, independent of CFTR genotype. So what does that mean? Basically, the genetic modifiers play an important role in whether you get diabetes, and also, quite importantly, the age at which you get diabetes. And some biology or background information on CF-related diabetes. It's associated with a decline in beta cell function, these are cells that make insulin, and also there's accumulation of amyloid in the islet cells of the endocrine pancreas. Insulin sensitivity is normal, which is unlike type 2 diabetes, but similar to type 2 diabetes, there's a family history that overlaps. So if there's a family history of type 2 diabetes in the family, then your rate of getting diabetes at an earlier age is increased in those that have CF, suggesting some genetic overlap between type 2 diabetes and diabetes and CF. Now, here is some genetic studies done a few years ago and then recently replicated. A genome-wide association study, a GWAS, for CF-related diabetes was performed and studied quite a few individuals. This one is a more recent one where 5,400 individuals were studied, and what we are looking at are the individual genetic variants spread across the entire genome. These are all the different chromosomes shown here, starting 1, 2, 3, 4, and there are literally millions of these genetic variants, and some subsets of those variants, as you can see, seem to show a distortion in their association with risk for diabetes. And there's a threshold above which we feel statistical significance has been achieved for all of these comparisons we're making. And one of those loci has a lot of these genetic variants in and around this gene called TCF7L2. Now that gene had been implicated in type 2 diabetes some years previously. And so now we've got a nice link between type 2 diabetes and diabetes and CF, and the idea that there are some physiologic or pathophysiologic overlap with type 2 diabetes. There's also genetic overlap. And that's seen with TCF7L2 and some other genes that are being explored by Scott Buckman and his colleagues at Hopkins, who have been performing much of this work over the past few years. Now, the other locus that brings us to A9 is shown right here. SNPs that were variants in and around this A9 gene were found to associate with risk for diabetes. So that's how we really focus our interest in this gene and its protein product. So where are these variants? Well, they sit right on the gene, actually, unlike many other GWAS studies where you sometimes find them at some distance from genes. In this case, the variants are either found just 5' of the gene or within the first exon. Now, the gene is shown in its orientation and the way we have it shown on a genome browser, which is actually in this direction. I know you'd want as usual when you're thinking of 5' to 3', but it just happens the gene is on the opposite strand and it runs the other way. It's just by convention, but it's easier to understand things when one goes back to the genome browsers if we show you how it's plotted. And the black dots here show you the genetic variants are associated with risk for diabetes, and there's a number of them you see here, including one in intron 5. Most of the rest are intron 1, the 5' end of the gene. Now, some of these variants have been associated with other traits in CF, and actually preceding the work of diabetes was work from the group at Toronto in collaboration with the International CF Modifier Consortium, which showed variants in this gene associated with risk of meconium also evidence of associated with lung disease severity, and work by Lisa Strug and her colleagues, work with data that had been generated by years prior from Peter Dury using immunoreactive trypsinogen, showed evidence that these variants also play a role in pancreatic exocrine function. So, and then finally, work by Marcus Moll and his group showing a 3' variant, 3' untranslated region variant implicated in asthma. So obviously, something's going on with this gene. There's genetic variants that are being implicated in several CF phenotypes as well as another pulmonary phenotype. So what role is it playing in CF, and what's the protein product doing? So the protein is very interesting as a candidate. Here we show a picture from some years ago that shows an interaction between CFTR and SLC26A9. A9 by itself functions as a chloride channel and reported to act as a bicarbonate and chloride transporter. Mentioned about the interaction, which is mediated by its C-terminal PDC binding motif, and also this STAS domain. And here in the picture, we show that series of interactions. So we have CFTR and A9 interacting. So again, furthering the plot here, or the theme, that the A9 is playing some role, could be playing some role in altering the function of tissues where CFTR is normally expressed. And of course, in the absence of CFTR, what's it doing? Perhaps substituting for chloride or bicarbonate transport. Now, more recent work by Dutzler and colleagues and others just published in Any Life in 2019, and then in 2020 showed the structure of this protein. It's a dimer, interacts via STAS domain. More recent work shows that its C-terminus modulates gating of this channel. I'll show you the picture of the picture, and colleagues who recently published in Cell Discovery also found a very similar structure. So it seems pretty firm that this is a dimer. And further studies of this show it's really a fast transporter of chloride. It doesn't seem to mediate bicarb transport much. So it's primarily a chloride transporter. And so, again, thickening the plot for its role in CF tissues. So the big issue, of course, is where is A9 and where is CFTR, and particularly in the pancreas. Now, initial thought was that if this is altering risk for diabetes, then maybe it's in the beta cells that make the insulin. Certainly been shown or it's been indicated that CFTR is in the beta cells, so maybe A9 is playing a role there. But studies that we did really changed our thinking about this, and were more in line with studies previously published by Lisa Strug, suggesting, and her colleagues, and suggesting that actually A9 may be playing a role in the exocrine pancreas as opposed to the endocrine pancreas. So let me show you that evidence. So here's a plot of the, or picture cartoon of the pancreas, and we're showing you the exocrine pancreas here, and sort of in these little stars here is the islet cells. They have alpha, beta, and beta cells and others, and these are the primarily the source of endocrine hormonal production, glucagon, insulin, and so forth. The rest of the pancreas is involved in producing pancreatic enzymes, and as you know, they're secreted into the duct through these acinar cells, and then that fluid that contains those enzymes and also a lot of bicarbonate, then transported down to the intestine. So that's sort of the lay of the land. So how do you find out where CFTR is expressed? Well, one way to do that has become more and more potent these days is to use single-cell RNA analysis. So you break up the pancreas into all different individual cells, you pull out a single cell, and you look at the RNA transcripts found in there. You ask yourself, well, where do I find A9 transcripts, and where do I find CFTR transcripts? The same cells, different cells, and what cell types are there? Well, the first thing you do is you cluster them to find out which sort of signatures, using what we call the transcription, they represent. And so you can cluster them in this fashion, and you can show that there are certain cells that have ductal attributes, others are ductal and acinar, and then a large fraction have acinar attributes. And this is human pancreas, or several of them, that was in a study that was performed by David Whitcomb and his colleagues at the University of Pittsburgh. And they were able to show these cell types as well as the islet cell types, the beta cells and the alpha cells and some other cell types. So where's CFTR? Well, there it is. CFTR is shown in yellow, and it's found primarily in the ductal cells and in the ductal acinar cells. Very little in the acinar cells in this study. And A9, shown in blue, is found in a small number of cells scattered throughout here, as you can see. And there's a number of cells that express both A9 and CFTR. That's great. The two transcripts are together. That means likely the two proteins are together in the same cell type in these ductal and acinar cells, not in the endocrine cells. Seems to be ductal. So, and then sort of further analysis shows here's the 15 cells where A9 was found in this one study, and the 500 cells where CFTR was found. There's an overlap of 11 cells where both were found, all in the ductal or ductal acinar. the rest of the A9 found in, again, ductal or acinar cells, and then finally CFTR, primarily ductal acinar, a very small fraction found in other cell types, which did include the beta cells, but primarily CFTR is found in the endocrine pancreas, as we sort of expect from studies of immunofluorescence and so forth, localization of CFTR. So that was the study that we did. Unfortunately, there were some very nice studies available. The raw data was available online, so we downloaded that data and reanalyzed it, and were able to confirm the finding. In 4 independent studies, the same was found. A co-expression of A9 and CFTR, which I'm showing in this box here. I'll just run down this column. So expressing cells were found expressing both 9 cells in this study by Barron and colleagues back in 2016, all were ductal. And it's pretty significant based on the number of cells that are expressing one or the other. 16 cells in this case, 28, and then 73, almost all ductal in this additional study from Segerslope. All these are pretty robust, significant associations. So I think we can say confidence for our own study with Whitcomb and colleagues, and then these 4 other studies all basically showing the same evidence that A9 and CFTR are expressed together in a small set of ductal cells, and CFTR is expressed primarily in ductal and acinar cells. So, okay, CFTR and A9 are together in some cells in the pancreas, in the exocrine pancreas, and ductal cells in particular. So how are these SNPs, how are these genetic variants that confer risk of diabetes, how are they influencing how A9 works? In fact, are they influencing how A9 works? Because some GWAS studies have identified variants that in fact are not altering the function or expression of the nearby gene. It's some other gene at some distance. But I think it was reasonable to start with A9, given the evidence I've shown you biologically, its role in CF tissues, to start thinking that A9 was likely to be implicated in this modifier effect. But I must say there is a possibility other genes might also be implicated in that same region. But we focused on A9 as a starting point. And we asked ourselves, well, what could the variants be doing? Well, might they alter in some way the protein is splicing? And that's because there's an issue with the first exon of A9 as to whether or not it's included in the full-length transcript or not, and I'll show you some evidence for that. So could it be affecting the splicing, the transcripts made, and shift the transcripts that are found, whether you have the variants associated with earlier onset or later onset of diabetes? What about something in the protein scene? Is it possible that the variants themselves aren't doing anything, but they're associated with, say, an amino acid change somewhere in A9, and that amino acid change is now affecting A9 function in some way? Possible, and so we wanted to take a look at that. And then finally, could it be that given that these variants are situated 5' of the gene in regions that are normally associated with regulation of expression, and certainly exon 1 or intron 1 could be also having expression elements, maybe it's affecting how much A9 is being expressed. So the components of A9 in the transcript, the actual sequence of A9, protein sequence, or something to do with the level of A9. So we went through those hypotheses. So first, here's our work on the transcripts, and it shows that there are 4 transcripts, 3 transcripts, sorry, available, or at least postulated to exist. My apologies, checking them out the time I had. There are 3 transcripts that postulated to exist, and they vary by the 5' region, whether or not there's an uncoding exon 1, form 3 is missing that exon, or whether there's this altered 3' exons. And so we did some detailed studies on human pancreatic RNA, and we can say that we could not find evidence of isoform 2 or 3 in the human pancreatic RNA. We used this 3' RT-PCR and 5' race. We looked also in stomach and lung, and we only found this one primary transcript. We also looked as hard as we could at these variants to see if they themselves might affect splicing. No evidence, and there's no evidence using this SPICE QTL, another bioinformatic approach, but we could find no evidence that these variants were altering the splicing forms of A9. and in fact could show there seemed to be one primary transcript in the pancreas, but also in the stomach and the lung. What are protein sequences? Some amino acid changed. And we sequenced the gene in 762 individuals and very deeply sequenced, found all the variants, and found two aggregations of genetic variants. Those that associated with high risk, 12 out of 12, and they found a cluster of 12 variants, and then another cluster of variants were associated with low risk. And these two clusters, these two co-associated sets of variants, which we call haplotypes, account for a vast majority of the combinations that occur in humans. So you have two primary or predominant haplotypes, and these haplotypes do indeed, as you'd expect, associated with age onset of diabetes, as they should. So I'm showing you homozygotes for the high risk haplotype, and you can see they have younger age of onset of diabetes, higher fraction at a younger age. If you're the low risk, you can see you have a substantially later age onset. And our analysis didn't reveal anything in the coding regions. No amino acid change, nothing, and no variants in coding regions that might change any other attribute of the protein, and no other rare or common combination of these SNPs. It seemed to be these SNPs were working as a group, 6 to 12 of them as a group, being transmitted together and affecting the risk for CF-related diabetes. So what about A9 expression? Given their location, here's a plot broken into 3 areas. Again, the gene is running from exon 1 through exon 2 in the direction I showed you before. Here's the location of the genetic variants in both the 5' region as well as the intron 1. There's, by sequencing, we can find elements of a basal promoter, the TATA and CAT box. So we know this should have promoter attributes. We can also find binding sites for transcription factors, and I show you them here. And in fact, when we take a look at the single-cell RNA-seq, and we take a look at the pancreas, and we take a look at the cells that express A9 and CFTR, these are the transcription factors found in those cell types. Unfortunately, those same transcription factors are found in PANC1 cells and CF-PAC cells. So we can use PANC1 cells and CF-PAC cells, because they have the transcription factors, they have likely binding sites here, to now test what the effect would be of taking pieces of DNA that contain either high-risk or low-risk variants and ask, do they affect the expression of luciferase, reporter expression system? And the summary is shown here. It's work done by Anpil in the laboratory and repeated many times to confirm it. There's a basal promoter, and the 5' end is indicated by the presence of the TAT box. SNPs have no effect. The couple of SNPs are here have no effect on how that basal promoter works. And there are 3 SNPs that are in this other region, but they don't seem to affect the expression of luciferase. In fact, there was no activity. There's no independent promoter. But when you put the two together, like putting an enhancer element working with a promoter, now you see both activity, an activity that is correlated with whether the SNPs are high risk or low risk. So there's a 12% difference in the expression levels if it's low risk versus high risk. And if you add in more SNPs, a few more from the 5' in this 5 kb region, now you get a 19% difference. A very subtle difference, but repeated multiple times. And then also repeated, the 2.3 kb results I show here were replicated independently in the CF-PAC cell line. So we think pretty good evidence for the following, that in the case of a CF cell line here, or a CF cell, a pancreatic cell, you have low risk variants. The presence of the phospho-Jun transcription factors will bind to those regions I just mentioned. They will produce more transcript, more protein, and perhaps with more A9 at cell surface, it can better alleviate the loss of CFTR function. In the case of the high-risk variants, and FOSA there, they bind, but you don't get as much expression of A9. And again, a reduced level of protein that's associated with the higher risk of developing diabetes and an earlier age of onset of diabetes. So with that, what are the implications for it? Well, modest differences, not surprising, modest differences in the level of A9 expression over lifetime seem to modify age of onset of diabetes. This was more substantial difference in expression, one might have expected a more pronounced effect on the phenotype. So it's not surprising that modifiers will have subtle effects because they're working lifetime in the patient with CF. As pointed out by others, and we certainly, our work supports it, A9 is a great target for CFTR agnostic therapy. And also, this emphasizes the potential for alternative pathways of chloride transport and CF treatment. And the last question is, well, how the heck does this chloride transporter in the pancreatic ductal cells in the extracorporeal pancreas, how's it really leading to a later age of onset? So there's a number of theories is somehow preserve islet cell number, change the inflammatory status of the pancreas, or to the number of functional islet cell progenitors. Ongoing work. I wanted to thank the people involved from my lab, David Wickham, pathologist who provide tissues, and Lowell Goff, our analyst, and also the funders and our international CF consortium. Thank you very much.